morning, dear students. Today we are going to learn about bile acids. Before going to understand about more about bile acid, let's see the names of some important scientists who were contributed some important finding relating with bile acids. Okay, so let's see. So bile salts are first crystallized by uh, Plattner in 1844, and the elementary formula for this cholic acid was reported by what is cholic acid? Cholic acid means it is the main bile, bile acid that present in the bile. So it is a most common bile acid that present in the bile. So its elementary formula was reported by Adolf Strecker in 1848, and cholesterol's elementary formula is correctly elucidated by Renitzer in 1888. So why we mentioned about cholesterol here? Because bile is forming from cholesterol. So that's why it's important to mention about cholesterol here. And the process of conversion of cholesterol to bile, it was demonstrated by Wintos, and for that contribution, for that finding, he got Nobel Prize in 1928. And a complete structural analysis of bile was done by Heinrich Wieland in 1918, and for that he got Nobel Prize in 1927. So next we are going to see more about the bile acids. These bile acids are synthesized in the liver from cholesterol. So from cholesterol, these bile acids are synthesizing inside the liver, and they contain 24 carbon atoms. So this is the structure of a bile acid, contains 24 carbon atoms, and all the bile acids have an alpha oriented hydroxyl group at position 7. This is the position 7. So all bile, that is a common property. All bile acid possess an alpha. What's orientation? Alpha oriented. Alpha oriented means that bonding, it projects below the plane of the ring. That I already told in some other video. Anyway, that OH is alpha oriented, as it is below the plane of the ring. That's why it is represented by using dotted line. So that is an important property of bile acid, the presence of the hydroxyl group at position 7. Next, we are going to see the process of synthesis of bile acids. That main important reactions that is happening during the synthesis is summarized as hydroxylation. That I already told you that from cholesterol that bile is forming. So the cholesterol undergoes hydroxylation mainly at the 3rd, 7th and the 12th position, depends upon the different types of biles. So first is that reaction is happening in that synthesis pathway. Then second reaction is removal of a 3 carbon unit from the cholesterol to make it a 24 carbon. So from that 27 carbon cholesterol, I need to remove 3 carbon unit to make it a 24 carbon. Because I already told you that the bile acid is consists of, that compound is consists of 24 carbon. So that is a second reaction. And third process is its conjugation with some amino acids like glycine. So by conjugating with some amino acid, that bile will become more soluble. So that property. So for gaining that property, it is conjugating with some amino acid. That is the third process. And after that conjugation, that bile acid is secreting to the intestinal canal through bile. So bile is a substance that is forming inside the liver, and it consists of so many other constituents in addition to that bile acid. So don't think that bile acid is the only component that present in the bile. Instead, In addition to bile acid, bile contains so many other things that I will tell you later. So, after the conjugation with the glycine, that bile acid is secreting to the intestinal canal through bile. And on reaching the intestine, again deconjugation, separation of that amino acid from that bile is happening, and also after that deconjugation, removal of the hydroxyl group that present in the seventh position, that also is happening. So that these are the important process. So next we are going to see all those reactions in detail, step by step way. So first is hydroxylation reaction. It is the rate limiting step in the process of the bile acid synthesis. It's the important reaction. That introduction of hydroxyl group to the seventh position. It is done by which enzyme? 7 alpha hydroxylase. 7 alpha hydroxylase. And it is a microsomal enzyme. Microsomal enzyme means what? Enzyme that present on the membrane of microsome. Microsome, and that microsomes are small spherical bodies that form because of the fragmentation of endoplasmic reticulum. So, 7 alpha hydroxylase is an enzyme that present on the wall, on the membrane of microsome. So, it's a microsomal enzyme. And that beta oriented, because how we could understand that this is beta oriented, because it is represented by a solid line. If it is an alpha, then it will be represented by dotted line. So here this beta oriented OH group that present on the C3 is converted to alpha type during this process, during this reaction, hydroxylation reaction. And that conversion of that beta oriented OH group to alpha, it is done by an isomerase enzyme. So we need to note the name of two enzymes here. This is the main enzyme that is adding OH group here, and isomerase is the other enzyme that is changing the beta oriented orientation of this bond to alpha oriented, to an alpha orientation. Next, a third OH group is adding at the 12th carbon here. In the case of a case if that bile acid is cholic acid is the main bile acid. And in the case of cholic acid, if the bile acid is a cholic acid, another OH group, a third OH group is adding to the 12th position. Okay, so this cholic acid, once more I am making it clear, it is a naturally occurring primary bile acid that represent major component of the total bile and total bile acid pool in human. So, if the bile acid considered is a cholic acid, in addition to the OH group present in third and seventh position, another OH also will added during this process at the 12th position. Okay. Next, we are going to see that reaction, that hydroxylation reaction happening to the cholesterol. This is the cholesterol, and we know that the enzyme that is acting on the step is 7 alpha hydroxylase. It's a microsomal enzyme. And in this step is one molecule of NADP plus H plus is utilizing and is converting to NADPH. And what's forming here? As an OH group is added to the seventh position, seventh carbon. So the compound formed is called 7 hydroxy cholesterol, 7 hydroxy cholesterol. And next I am going to mention about another bile acid, that is cholic acid, another primary bile acid. It has two hydroxyl group at position 3 and 7. It doesn't possess the hydroxyl group at 12th position. At the 12th position also, an OH group is present only in the case of cholic acid. The case of another bile acid, that is quinodeoxycholic acid, it possesses only two hydroxyl group. In which all position? Third and seventh position. So that structure also we can see it possesses only two OH group at the third and the seventh position. Then we are moving to the next second step, that is the removal of a 3-carbon unit. So what all processes are taking place during that step? The side chain is first hydroxylated at the 26th carbon. This is the 26th carbon. 26th carbon. It undergoes hydroxylation. So hydroxyl group is added, and then the same point, that carbon itself undergoes oxidation. And what is formed here? COH group. COH group get added there, the same 26 carbon. And after this addition, a cleavage happened to here. This is the 24th carbon. 24th carbon. There a cleavage happen, and by that way a 3-carbon part is removed from the whole structure. By that way a 24-carbon compound primary bile acid is possible to form here. Next, we are going to see the formation of bile salts. The one that has formed now is primary bile acid. So next, that primary bile acid that is formed because of the removal of that 3-carbon compound. Next, it combined with some amino acid. That's the process happening here. Amino acids like glycine or taurine. As it get combined with this type of amino acid, then we can call the primary bile acid as conjugated. Okay. Uh, then depends upon what type of amino acid get connected combined with the bile acid. There exists different type of conjugated bile acids like glycocholic acid, taurocholic acid, glycoquinodeoxycholic acid, likewise. And among the different conjugated primary bile acid, which is the main conjugated bile acid, is glycocholic acid. Why? Because in the beginning of the chapter itself, I told that the predominant bile acid in human bile acid pool is cholic acid. So, as it is the predominant bile acid, there is most chance for this one would be as the major conjugated bile acid too in the bile. So, understand simple. Understand in this way, glycocholic acid is the major common conjugated bile acid. Its picture is shown here. And in this picture, we can see this cholic acid get conjugated with the glycine. So by that way, it become conjugated. Then what is the purpose of this conjugation reaction? By undergoing conjugation, it will get attached to so many polar groups, and get attached with so many polar groups, its surfactant property become more efficient. Surfactant means where these are substances when get added to some other thing, it increase its solubility. So that's what conjugation increase or improvise the surfactant efficiency of bile acid. Then after get conjugated, the bile acid is excreted through the bile. Okay, and as it excreted through the bile, it exists in the form of bile salts. Bile salts means it contains some sodium or potassium salts. Potassium salts along with that conjugated form. So, in the bile, it exists as bile salts. Why it is called bile salts? Because some sodium or potassium are also coming and bind with that conjugated bile salts. Next, we are going to understand how the primary bile acid get converted to secondary bile acids or bile salts. So on reaching the intestine, the primary bile acid is acted upon by intestinal bacteria. What these intestinal bacteria are doing to this primary bile acid? They separate, they remove the amino acid from the primary bile acid. They remove that conjugation. So, deconjugation is taking place inside the intestine. It is done by the intestinal bacteria. Okay. And after the deconjugation, then one more thing is happening to that primary bile acid, to making it to a secondary bile acid. What's that second thing happening to this primary bile acid? That is, we have learned that at the seventh position, there present a hydroxyl group. So that hydroxyl group is removed. And after removing that hydroxyl group, then that primary bile acid become secondary bile acid or secondary bile salt. So in that way, cholic acid is converted to deoxycholic acid and ketodeoxycholic acid is converted to lithocholic acid. Let's see the structure of a cholic acid. This is the cholic acid. We can see the OH group at the seventh position and that is removed and as a result form a deoxycholic acid, that is a secondary bile acid. So in this way, bile is synthesizing. Okay, I hope that's clear to you. With this, I'm going to finish today's section. Thank you.
I'm an icon, man. Ain't my girl about a 10. I should fuck her in the Benz.